I'm Professor Matthew Schmidt, and this is Genetics. In today's session, we're going to ask a question which almost sounds overly simplistic, and that is, what is the function of a gene? I hear some of you saying, well, isn't that what we've been talking about the whole time? I mean, a gene controls a trait, right? Well, that may be true and it may not be true, but given our newfound, in the context of this course, knowledge about the structure of DNA and the fact that somehow DNA is what genes are made of, we really need to ask ourselves a couple of questions and say, yeah, Mendel, implied that one gene controls one trait, but what does that really mean at the more molecular and biochemical level? And certainly we're not going to quite get there yet, but we need to explain, well, we're going to start to get there, how the gene, whatever it actually may be, in fact controls the phenotype in reality. So this is part of the section called Mutations and the Nature of the Gene. We're really going to get at the nature of the gene in this section. We're going to use some of the knowledge that we learned about mutations last time in order to be able to do this. So there's a really landmark set of experiments that were published in 1942. If you'll think about when that was, it was a full 14 years before Watson and Crick actually published their structure of DNA. So this was understood, it was sort of going together at the same time. It's so important to realize that structure and function are so intimately related, but there are different ways of getting at structure and function. So while Watson and Crick and others were working on DNA, structure, chemistry, etc., it's also important to be working on the function. Ultimately, the two have to be able to mesh together perfectly, right? So these experiments, it seems like we're going back in time, but in a sense, these were two trajectories that eventually are going to come together, all right? So two very famous scientists called Beadle and Tatum did these experiments, and they performed experiments using the mold Neurospora. I think we mentioned it once before when we were talking about mapping centromeres. And really, I'll probably get very overexcited about this, but this really revolutionized our idea about how genes work. Remember, the key thing here is function, not so much structure. Sometime we're going to have to be very clear about how the structure relates to the function. But it turns out that Beadle and Tatum were interested in the biochemical pathway. They were using this pathway that allows synthesis of the amino acid arginine. And they were also interested in the relationships of genes to this pathway. So without any genetic knowledge, biochemists knew that there was a pathway, and by the way, you may know this, many biochemical reactions occur in these multi-step sort of pathways, and they can be worked out, I'm not going to say easily, but it's certainly done, and a lot is known about it. So basically they said this: if you take Neurospora and you grow it on what we call a minimal media, which means they're just the real basic precursors of things there, they can grow, but they're doing a lot of things. But what's relevant to this pathway is they can start out with some precursor molecule and ultimately build it into arginine, which is an amino acid. I'm sorry if I already said this. All living things need arginine. Humans, as an example, we can only synthesize roughly half of the amino acids we need. The other half we have to consume. Organisms like this can probably make all of them, but one thing is for sure, they know how to make arginine. It was also known that this process actually took place in a stepwise fashion. So some precursor molecule, which you don't have to really care what it was, what really was going on was that was being converted into a substance called ornithine. If you want, you could almost think of that as a pre-amino acid. That ornithine was getting converted into citrulline, which is another sort of pre-amino acid, and ultimately the citrulline was being made into arginine. So this was known. Furthermore, it was known, you guys probably know this already, isn't it true that every biochemical reaction requires an enzyme to both facilitate and control that reaction? Yes, it is true. So right now we don't need to worry about the names of the enzymes, but the bottom line is that what we've sort of arbitrarily called enzyme X here was responsible for this reaction, making the precursor into ornithine. Then an enzyme we're going to call Y was responsible for that next step where ornithine was converted into citrulline. And finally, enzyme Z allows the citrulline to be converted into arginine, which is the end product that's necessary. So far that's what they knew. But then they got into some genetics, and we're going to combine genetics and biochemistry here. So what they did was, now we're still trying to get at gene function. They mutated spores of Neurospora and isolated various auxotrophic mutants. We used that term once before. An auxotroph is an organism that is not able to synthesize something that a vital nutritional compound that the wild type can. So auxotroph means that it needs to be fed that particular item in order to, it has to be supplemented basically in order to grow. The opposite of an auxotroph is a prototroph, just in case you want to get that terminology down, a word which literally means before the feeding. So in other words, prototrophs can make everything they need to grow before they get fed or supplemented with things in the media. So auxotrophs, they just started basically collecting mutants that they found could not make arginine after they had mutated them. I don't remember what they did. Maybe they irradiated the spores. We talked about last time the various ways that you could induce mutations. So they just started cataloging these mutants. They called one Arg minus number 1, Arg minus number 2, Arg minus number 3. They collected a whole bunch of them. So obviously I'm going to be condensing this data and presenting it to you in a much simpler fashion than they did. But it turns out, so look what they did. They're causing genetic mutations, clearly, and they're finding out that some of these mutations disrupt somehow this pathway that they know to be occurring. And how do we know that? Because they can't make arginine anymore. One of the things that they did was to show that if they added arginine to the medium, they could grow. So with arginine around, they're good. Without it, they're not. Thus, auxotrophs. Now, they did a lot of stuff that we're not going to talk about, but in particular, there were 3 mutations, which we're going to call Arg minus 1, 2, and 3, and they did some genetic mapping of where the mutations existed by some similar methodology to what we've discussed in the past, and they found something really exciting and interesting. All 3 of these mutations were mapped to 3 different chromosomes. Neurospora has a lot of chromosomes. What this means, though, is since we know that a gene is located on a chromosome, this means that even though the phenotype was the same of all these, what's the phenotype? It's that they're Arg minus. They don't make arginine. But these 3 mutations, all which had the same phenotype, had problems in 3 different genes based on the mapping evidence. Now, there's a table here that shows some growth data for these 3 different auxotrophs. This, I've seen people get confused about this data before. I think it's mainly just a matter of reading it correctly. So let's just take a minute to make sure we understand what this is saying, okay? So here are the 3 different auxotrophic mutants, Arg minus 1, minus 2, and minus 3. This O stands for ornithine, this C for citrulline, and this A for arginine. So basically, this is what it means, okay? If you take the mutant Arg minus number 1, here's the deal. If you give it arginine, we know that that's going to allow it to grow, right? And you see that plus there means growth occurs. But interestingly for this one, look, if you give it citrulline, it also can grow. You may be seeing what this means, but stick with me. If you give it ornithine, it'll grow. That just means that you can supplement it with different things along in the pathway. So we're building something here. Just stick with me and then we'll put it all together. Conversely, Arg-2, that particular mutant, if you give it ornithine, look what it happens. It cannot grow, so there's a difference there. However, if you give it citrulline, it can go ahead and grow, meaning that eventually, whatever you're giving it, you have to get the arginine. If it grows, that means arginine's being made, right? And of course, it makes sense that if you give it the arginine, any of these mutants are going to be able to grow because that's what they need. Lastly, if we look at Arg- 3, if you give it ornithine, it's not happening. Give it citrulline, no good. Of course, we know if you give it arginine, it's going to be able to grow. So guys, what does this mean? It's just so important. I'm bursting with enthusiasm. Now, on the next slide here, I've kept the data, and I also put the pathway there so we can sort of compare them and see what's going on here. So the reasoning of Beadle and Tatum at this point was that they said, look, each of these different mutants is blocked in this pathway, but they're blocked in a different position. So let's look at the data and look at the pathway and see what we can come up with here. We can almost be as smart as Beadle and Tatum were. Probably not, but almost. All right, so think about Arg-1, right? If you give it ornithine, it can grow. Does that not imply very strongly that it can do this and it can do that? Because that's the only way to make arginine. The pathway is always the same, right? So if you give it ornithine and it can grow, it strongly implies that enzyme Y works fine and enzyme Z works fine. Let's see if the other data is consonant with that. If you give it citrulline, it can grow, right? Well, that's because, yeah, all it has to do is that last step. And of course, if you give it arginine, that doesn't really give you a lot of information. So what they reasoned was that reason that Arg-1 mutant was in fact a mutant, was an auxotroph, Arg-, was because it had a problem with this enzyme X. In other words, it had an inability, I'll sort of draw lines through it, it looks a bit odd, but that means that's the reaction it cannot perform. It can't get the precursor and turn it into ornithine. I hate to be redundant, but I need you to understand this. Because if you give it ornithine, everything goes along just fine. You give it citrulline, everything goes along just fine, etc. So they said the problem here resided with the enzyme X, all right, in that particular mutant strain. Let's sort of carry on this analysis with the others. So with Arg-2, if you give it, I think it would be a good idea to erase this stuff. Let's start over for this analysis. Arg-2, if you give it ornithine, it's not working, right? That strongly implies that this is the problem right here. Because if you give it citrulline, it grows, right? Which means this step is okay, so enzyme Z is all right. So it's strongly, and arginine, we don't have to keep saying it. It implies that there's a problem with the enzyme Y in this one, in Arg-2. Now, some of you may be thinking, look, there's no way to prove that the enzyme X is working in this strain. However, if it made the precursor and turned it into ornithine, that's the same as giving it ornithine in a sense, right? And we see that if you give it to it, it can't do anything with it. So this gets a little further than we can go, but we need to make the assumption that there's one thing wrong with each of these mutants. And in that case, it's the enzyme Y that's broken, if you want to think of it that way. And lastly, we'll erase this again. In the case of Arg-3, what do we see? We see that if you give it arginine, of course, we know that it's going to work. But if you give it citrulline, it's not working, which strongly implies this step is the one that cannot occur and that the problem is with the enzyme Z, right? Because now, again, it's the same thing. I can't prove that both these enzymes are functioning, but if they were, you'd end up making citrulline, and we know that if you have ornithine or citrulline, it doesn't matter because this one is blocked in this last step. So they basically said, even though none of these strains can make arginine, they're all auxotrophic mutants, it's for a different reason. And what I think what you're about to find out is they reasoned that ARG1 was blocked in the pathway at step 1, which is controlled by enzyme X. ARG2 at step 2, which is controlled by enzyme Y. And ARG3 at step 3, which is controlled by enzyme Z. So this is so important. What they concluded was that there are 3 different genes mutated in these 3 strains. The mutant genes cause the production of enzymes that don't work, non-functional enzymes. And therefore, dun-dun-dun-dun, the purpose of a gene is to produce an enzyme. Now, let's just pause for a minute. This is big stuff. If in the beginning you said, well, yeah, gene controls a trait, Mendel said that. Yeah, but Mendel didn't know anything about enzymes and genes and the pathways that were causing production of a pigment. This is really the first evidence that says at the molecular level, there's a relationship between, they didn't say DNA, they said gene information, right? The information, whatever it is, well, what that information does is it tells a cell how to make an enzyme. If that information is damaged or mutated or changed, then the cell no longer can make that enzyme, and it will show a phenotypic, in this case a problem, but a phenotypic difference because of that, right? Now, they did all sorts of experiments like this. It was so powerful that they came up with this idea that you may have heard of. Again, remember, this is all before DNA's structure was really worked out. They called it the one gene, one enzyme theory. It's huge. So basically the summary of it is that biochemical pathways tend to occur as a stepwise series of reactions. Each reaction, this part's not revolutionary, right? Each reaction is specifically catalyzed or facilitated by a very definite enzyme. And the culmination here of what we want to get across, and what they wanted to get across, is that each enzyme is synthesized somehow, we're going to have to get to the how later, by a specific gene. One gene, one enzyme. Now, I can't overemphasize how vitally important that realization was, even before it was known that DNA was structurally, well, I should say this was around the same time as Avery, McLeod, and McCarty were really doing the experiments that showed DNA probably really was the genetic material. But remember, 2 different pathways. One was more sort of functional, one was more structural. To update it a little bit, we now know that there are enzymes that consist of more than one polypeptide chain, and it turns out that a gene actually codes for one polypeptide. So if you wanted to make it a little bit more specific, you could say instead of one gene, one enzyme, one gene, one polypeptide. And this also further recognizes that while they were working with enzymes, enzymes are proteins, right? So really, it's a relationship between the gene and the protein which has some function, be it enzymatic or be it something else. Now, I guess if you haven't got it yet, this was a major, major discovery. And it finally allows us to understand what a gene really does. What does it really do? It makes some kind of protein somehow. And also it tells us what the relationship between genotype and phenotype is, at least in a rudimentary way. Because what it means is this. Phenotype is proximally determined. Proximally just means directly determined by particular enzymes or proteins that exist in a cell. And an organism, if it's multicellular like us, your phenotype is the net total of the phenotypes of all of your cells. It's a little odd to think of it that way, but it's true. Now, basically, at the risk of redundancy, let's just make sure we understand this. So genes, we're saying, make certain proteins, and those certain proteins are specifically and proximally responsible for the phenotype that comes out. Now, if those genes are disturbed, the proteins will be disturbed, and that may alter the phenotype. Often not in a good way, but that we're not needing to make a value judgment. Now, since enzymes are generally speaking proteins, it's certainly for our purposes, it's good to just make it simple like that. What really turns out, it's getting more and more exciting. I don't know how you guys can even stand it. You're probably, I can't see you. I guess you know that. You're probably jumping around and all sorts of things like that. But it turns out that how a gene builds a protein, we're getting closer and closer, is that a gene specifies the sequence of amino acids in a protein. That's really ultimately what makes one protein different from another, right? There's 20 different amino acids and they get lined up in different ways, and that's what makes a protein, one protein different from another. So first, the way we approach this is I first gave you the evidence of Beadle and Tatum, then we got to one gene, one enzyme. I'm sort of doing this backwards. Now I'm telling you that genes specify the sequence of amino acids. We're going to get to the evidence for that in a moment, but I just want to tell you, because we're on a roll here, that it's true. So various experiments established what we refer to as gene-protein collinearity. What that means is that this linear sequence of nucleotides in DNA, A, G, T, T, C, whatever it is, correlates in a linear fashion with the sequence of amino acids in a protein. The gene nucleic acid sequence encodes or determines the protein's amino acid sequence. Huge. Now, before I tell you how this was actually discovered, let's just look. We can see things in a new light now. So many human genetic disorders are caused by a single gene mutation, which causes, we now know, a single enzyme or other protein to alter its function, which, it's not always a disease, but in this case I want to look at the ones that are. And I also note that these conditions are usually recessive. Now, why would that be? Maybe it's obvious, but since we're diploid, right, we got two copies of a gene in the cell. If one of them is broken, for lack of a better word, you still have the other one working. So if the point of these genes is to make a particular enzyme, as long as one of them's making it, it's probably able to fulfill its function, even if the other one is silent. It's when both of them are broken when you have a problem, which is what recessiveness really means, right? So let's take a couple of examples that some of them I think we've looked at before. I know we mentioned PKU when we were talking about human genetic disorders that are transmitted in a Mendelian fashion, but now we can get a little more specific about it. So that meant that one gene was responsible for PKU. Now I can tell you that the gene that causes PKU, when it's mutated or altered, encodes an enzyme called phenylalanine hydroxylase. Now this is in the biochem class, but basically that enzyme is in a pathway that it converts phenylalanine to tyrosine, which is another amino acid. It doesn't work. It can't do that. So look what happens. Phenylalanine builds up, which can be dangerous, and it's hard to make tyrosine, which also is not the best thing. This is a potentially life-threatening condition. Now that we understand it, dietary restrictions, you can't eat a lot of almost any phenylalanine, can allow somebody to live pretty much normally, but it has to be caught early, and that's why it's so important to understand these things. Just for fun, if anyone's drinking a diet drink or you have access to one, aspartame has phenylalanine in it. It's 50% phenylalanine, so by law it has to state on there. It'll say right there, I don't know if you ever noticed it, phenylketonuria, this product contains phenylalanine. Don't drink it, in other words. So that's one thing phenylketonuria can have, among others. To extend this, it's actually in the same pathway. Albinism, we wouldn't call it disease, but it's a condition where someone, and it doesn't matter what ethnic background you are, if you have no melanin in your body, you're an albino, right? Albinism is a problem with the next enzyme in this pathway, which is tyrosinase. Now, it's a lot more complicated than this, actually, if you look it up. But tyrosinase takes the tyrosine and allows it to be converted to melanin. There are a lot of intermediate steps, but the bottom line is, without functional tyrosine, you don't get melanin, and so this is in the same pathway, one gene, okay? Tay-Sachs disease is definitely a serious disease. The enzyme, it's a bit of a tongue twister. It's hexosaminidase A is affected. I This is where the biochemistry gets, you can look it up, but basically this enzyme is responsible, number one, it exists in lysosomes, and it's responsible for the breakdown of certain things called gangliosides, which have a lipid component to them. Now, it's known as a lysosomal storage disease because since they can't be broken down, these products build up in lysosomes, particularly affect the development of the nervous system, and usually this leads to death by age 4. This disease has been pretty seriously studied. It is almost uniquely confined to a certain Jewish population, Ashkenazis. I don't know if I pronounced that properly, I apologize. But these are Jews of European descent who tend to basically, how can I say it, not breed a lot with other people who are not also of the same ethnic background. So that's mutation that occurred, presumably in that line, has not really spread out to a lot of other ethnic groups, which is interesting. So if two people who are of that background are going to have children, genetic testing for this and some other conditions is usually indicated. Now also, we already said it, but I just want to prove to you, it's not just about enzymes. There are other genetic conditions that affect non-enzyme proteins, and that really shows convincingly that genes, it's not just one gene, one enzyme. It's one gene, one protein. We've already said really one polypeptide. But for example, in sickle cell anemia, there's a problem with hemoglobin. It folds improperly, it causes red blood cells to take on an aberrant shape, and that hemoglobin, that's the one protein. Hemoglobin is actually made up of 4 polypeptides of 2 types. So the problem is in the beta subunit, the gene, the protein. Cystic fibrosis is a potentially life-threatening disease. It's a problem with a protein that has a ridiculously long name called the CF for cystic fibrosis, transmembrane conductance regulator protein. Fancy as that is, it's basically an ion transport channel. The bottom line is there a lot of epithelial functions are affected and breathing becomes problematic. I mentioned xeroderma pigmentosum in the last session. You get the same phenotype if there are problems with various genes in this nucleotide excision pathway, in which case many of them are enzymes, some of them may not be. But the bottom line is, I guess you figured it out. Genes make proteins. Mess up the gene, more than likely you'll mess up the protein. At least you'll change it. Not always does it have a big deleterious effect. I mean, for example, we're talking about diseases, but if we go back to Mendel's pea plants or our eye color, there's an enzyme that works to make melanin to give people brown eyes. And I'm going to oversimplify it a little bit, but let's just envision that the big B allele makes that enzyme. The little b allele makes a different enzyme, which makes less amount of melanin in people that have blue eyes. So in that case, having different alleles does lead to different phenotypes, but you wouldn't call one a disease state, right? Now, I told you that I would give you the evidence for this, and it's really rather important. You could really get into this. This guy, Vernon Ingram, studied sickle cell anemia, among other things. And this guy is actually known as the father of molecular medicine, because he was the first one to take a disease condition and really show it's sort of all connected in with Beadle and Tatum in a way that you'll see. But they were more pure researchers. This guy was more into, he was a medical doctor, I think. So clearly, I mean, sickle cell anemia is a genetic trait. It's inheritable because, how did he know that? Because he saw that it runs in families. And he showed, in fact, that hemoglobin was the culprit. So hemoglobin consists, as I said, of 2 alpha polypeptides and 2 beta ones. And he did a lot of work sequencing the hemoglobins from healthy people and those with sickle cell anemia, and was able to show that the hemoglobin was different. In particular, there's one amino acid that's different. Now the amino acid valine is replacing at position beta 6 the amino acid glutamic acid, which is the correct one. The point of the logic here is people with sickle cell anemia have a problem with their hemoglobin, and it's because one amino acid has changed. Subsequently, it was shown that the cause for that change was a one nucleotide mutation in the gene. So the gene has a problem, now the polypeptide has a problem. Sadly, now the person has a problem because hemoglobin is not functioning properly. So this is really what established the gene-protein collinearity. You look at the gene, there's a problem right there. You look at the protein, there's a problem in a similar linear position. And that's the bottom line, guys. The linear sequence of nucleotides in a gene corresponds to the linear sequence of amino acids in a protein. This is getting too exciting. So we're almost done with this part, but eventually we're going to really have to address the mechanism, right? It's always good. We're building in steps, but each answer yields another question, which is, okay, great. How the heck does a gene tell a cell how to make a protein? We have some clues here, but we don't know the actual mechanism. So quickest review of protein chemistry, because if we need to be able to understand this in order to understand how genes deal with amino acids. So just remember these basics, and if you need a little refresher, any regular bio or chem or biochem book should be able to help. But remember that proteins are polymers of 20 different amino acids, and they fold up, which is very important for their function. So the so-called primary structure of a protein is the sequence, the linear sequence of amino acids. That's directly what a gene controls. Once that's settled, that protein will begin to fold into secondary and tertiary structures, sometimes known as higher order folding. And a protein only can function if it's folded up correctly. If it's not, you've probably heard the term denatured. It literally means that the protein is not the way it's naturally supposed to be, and often that means it will not be able to function or function properly. And also remember, quaternary folding refers to the phenomenon where we have a protein that consists of more than one polypeptide, and folded polypeptide chains associate with one another to form the functional protein, like in hemoglobin. Well, it's getting so exciting, and that's a lot to digest, so go ahead and digest it, and I'll see you again real soon.